third lecture on Chapter 5. This one covers enzymes. What are they? All right. Enzymes are proteins. Let's begin by describing their properties. So one property is that they are made out of protein. Some enzymes are not made out of protein. Some. I mean, they're kind of the exception to the rule. RNA, for example, has been shown to act like a catalyst, speeding up reactions, just like proteins do. But by and large, the vast majority of enzymes are proteins, and so that's why you'll see your textbook describe enzymes mostly in the form of a protein. A second feature or property or characteristic of an enzyme is that they are catalysts. If you haven't heard of the word, in chemistry this means that they speed up reactions. All right, so R, X, and S is my way of saying reactions. Okay, same thing. How do they do that? We're going to talk about that in the next few slides, so let's just hang on. A third property of enzymes is that they are highly specific, meaning that you need one enzyme for one specific reaction. You would need a separate, totally different enzyme to do some other different reaction, and you would need a third enzyme to do a third type of reaction, so on and so forth. We have so many reactions, we need separate enzymes for each one of those. You cannot use one enzyme to do every single reaction of the body or of a cell. You need specific enzymes for specific reactions. And 4, enzymes are reusable. What this means is that they don't break down once the reaction has been completed. An enzyme keeps working over and over and over and over and over again. This is what's great about them. Okay, so let's stay on this for a little bit. Let's say that my job as an enzyme is to take a blue marker, right? See the blue markers that I use in lecture. And my job is simple. My job is simply to uncap, right? To uncap markers. That's my job. So whenever I uncap them, that's basically like breaking a bond. And that means energy gets released, right? If you keep giving me black markers, I keep doing the same exact job of uncapping them, releasing their energy, because I am reusable. But then one day, somebody gives me a red marker. And I might grab that red marker, I might try to uncap it, but I realize that I can't. And I can't uncap it because I am highly specific. I only work on blue markers. You would need a different enzyme to uncap this red marker. Okay, so I hope you understand the difference between being highly specific, right? And that you understand what's meant by being reusable. All right, we're going to tackle the catalytic property of enzymes in the next few slides. All right, so let's get to it. All right, we should be able to recognize this sort of energy scheme because we saw it in the energy lecture. All right, so here's my graph, energy on the y-axis, and on the x-axis, I have the progress of the reaction. Notice that the reactants have a lot more energy than the products. And notice that the reactants don't become product instantaneously. And that's because there's this massive energy barrier that you need to overcome. And that energy barrier is called the activation energy barrier, or the energy of activation. Oftentimes we'll see it denoted by an E sub A, the energy of activation. So what is that? I don't want this to be mystical, but In this case, we're looking at sucrose. This is, if you remember, table sugar. And sucrose is made up of glucose and fructose, two monosaccharides attached to each other to form a disaccharide. And the bond right here between the two is stable. It doesn't break down easily. Therefore, this table sugar is going to stay like this for years and years and years, right? Glucose connected to fructose for years. But we want to tap into the energy of this disaccharide, so we are invested in breaking that bond. How exactly is it that an enzyme helps us break that bond? Well, remember that stable bond here? What an enzyme would do is it would take glucose and fructose, right, the things that make up sucrose, and it would destabilize that bond. It doesn't necessarily break it, it just makes it a little bit weak so that the amount of energy that you used to require is now less. And for that reason, which case, A or B, is most likely to happen the quickest? Well, it would have to be B, right? Because there's just a smaller wall to have to get over. And do you notice that the bond is now broken? And that should mean something to you. Breaking a bond means energy is released. That means that this is an exergonic process. And that's about right. Enzymes perform exergonic reactions. All right. So enzymes lower the energy of activation. They do not give you energy to speed up a reaction. They simply lower the amount of energy required, right? So let's do some sort of silly analogy here. What if you saw a coat? Can I draw a coat? Well, I don't know. No, the answer is no, I can't draw a coat. But I'm going to try anyway. Oh, so far so good. Yeah, oh, it's not bad. Oh, it's going to work out. Okay, it's more like a shirt. All right, so you found a shirt that you really want, and that shirt is like $500. I don't know where you would find this shirt, but that's what it's worth. And unfortunately for you, you don't get paid until the first of the month, and the sale is going to end on the 25th. That means you're going to miss out on this really, really cool, expensive shirt. So you go to your friend Enzy, and you say, Enzy, help me out. I want to get this shirt. I want to get this shirt quickly. Can you speed that up for me? What does your friend Enzy do for you? Remember, enzymes don't give you energy. So your friend Enzy cannot loan you the money so you can get this shirt. Enzymes instead lower the energy of activation. So what does your friend Enzy do instead? Your friend Enzy goes to the store owner and says, hey, listen, you know that shirt you got for $500? I want you to sell it to my friend for 1/5 of a penny. And you walk out of that store with your shirt. And I don't think I've done justice to just how amazing enzymes are. But I think you sort of get just how enzymes might be pretty amazing. All right, so one more time. Here's a reactants, here's a products, right? We saw this sort of graph before. This is definitely an exergonic process. Why? Because you start out with a lot of energy and then you end up with products that have lower energy. So you must have released energy in this process. All we're saying is that enzymes will help you get that energy faster by simply lowering the amount of energy required to get over that little hurdle, the energy of activation. All right, let's take a look at the mechanisms, right? How do enzymes perform these tasks? So I'm going to draw my enzyme here. I like to draw it like a blob, give it a little pocket there. And then I do one of these jobs. All right, that's my enzyme. I'm going to call that pocket right there the allosteric site. And I'm going to call that pocket right there the active site. All right. Okay, so what is the active site? The active site is where the reaction takes place. All right. And the active site actually determines how specific an enzyme is. So here is a molecule that looks like it can fit in there. So the molecule fits in there, right? And we can give it a color. All right, so there it is. It's inside the active site, and now what can happen is we can spit out a product, and the product will look different, right? All right, so this is my reactant. This is my product. Now, because the reactant cannot become a product unless it's in the presence of an enzyme, reactants get a brand new name, and the name is substrate. And that's what you're seeing here, all right? So from here on out, if you hear the word or come across the word substrate, you should look for an enzyme. And if you come across the word enzyme, you should look for a substrate, okay? And so how can you look for these things? You know, are there certain clues? And the answer is yeah, there are. For one, enzymes have a tendency to be named after their substrates. In fact, enzymes are given a two-name system. One of the cool enzymes you'll learn about in this unit and in lab is an enzyme by the name of catechol oxidase. Catechol oxidase is an enzyme responsible for the browning of things like bananas, apples, avocado, potatoes, eggplant, right? I'm sure that if you spend any kind of time in the kitchen, or even if you don't, I mean, if you come across these fruits or vegetables, you're going to see that they turn brown as soon as you slice them open and expose them to the air. And that's because of this enzyme, catechol oxidase. The enzyme, catechol oxidase, is named after its substrate, catechol. And the second name tells you what the enzyme did to catechol. It oxidized it. And notice that the enzyme has an ASE ending. All enzymes have an ASE ending. So if I say something like alcohol dehydrogenase, I got to work on my penmanship here. Alcohol dehydrogenase, you would know, is an enzyme because it ends in ASE. And you would also know what its substrate is. What's its substrate? Alcohol, that's right. Because all enzymes are named first by what they work on. And that's the substrate. So alcohol dehydrogenase works on alcohol. What does the second name tell you? It tells you what the enzyme did to alcohol. And in this case, it dehydrogenated it. It removed a hydrogen from it. And there it is. That's how you will be able to navigate through the lingo of enzymes and substrates. So let's take a look at another enzyme with a shortened name. It doesn't have the two-name system that I just explained, but it's an important enzyme nonetheless. In this case, we're going to look at the example of the enzyme sucrase. All right, see, still it ends in ASE, so that's always going to be there. That's going to be very helpful. And so when you read sucrase, you know that you're dealing with an enzyme, and you know to expect a what? You know to expect a substrate, right? These two terms are always used together. The enzyme sucrase has an active site that is in the shape of the substrate. Again, that's what contributes to this idea that enzymes are specific. So once the substrate enters the active site, the enzyme then gets busy destabilizing the bond between glucose and fructose. Here's the shape of the bond right now, and look at the shape now of the molecule that's being destabilized. And now notice Water is entering the equation. You need water to break that bond. That should be a big clue that this kind of reaction is called hydrolysis, right? And you should remember from the energy lecture that hydrolysis is associated with exergonic reactions. These are reactions that release energy. Remember why they release energy? Because by breaking a bond, right, where energy is stored, the bond here is missing. And now glucose and fructose are no longer together. And once you broke that bond between the two, not only did you release them from their attachment, but you also released a little bit of energy. Later, in respiration, we'll learn that we have enzymes that break down glucose and fructose and release even more energy. Okay, I'm hoping that you understand why enzymes are really, really important to us, right? At least on an energy level, they are the ones that are making it easy for us to deliver energy to all of our cells. And we have trillions of cells, and we have a very active body. We need that energy right away, and we couldn't be able to tap into the energy of something like sucrose because of that bond right there being so stable. Fortunately, our enzymes destabilize the bond right here, use water in hydrolysis to help break up that bond, releasing the energy, and then bunches of other enzymes contribute to the further release of even more energy. All right. I'm hoping that all my students will be able to explain to me how enzymes speed up reactions. I'd like to see them explain to me this energy scheme of how reactants become products, right? And how an enzyme lowers the energy of activation so that the reactant becomes a product a whole lot quicker. Let's discuss how you might inhibit enzymes, right? There's essentially two ways, and then feedback inhibition, which will resemble one of the two that we discuss up here, competitive and non-competitive inhibition. Competitive inhibition is carried out by chemicals that resemble the substrate. And because they resemble the substrate, that means that they can also fit into the active site. And because they can fit into the active site, once they're inside, they block the natural substrate from getting in. And so that's why we say that they are acting like competitive inhibitors. So I'm going to draw an enzyme, again here. There's my one pocket. There's the active site. And here's the natural substrate right here. Right? But what if one day the enzyme comes across something that looks like this? Right? Notice that they're not exactly the same, but boy, are they similar. This is the natural substrate. This is the competitive inhibitor. Now, what if the competitive inhibitor were to enter the active site? Well, it would sit here, right, with that little leg that the natural substrate doesn't have. And as long as a competitive inhibitor is in there, who cannot go in? The natural substrate. And therefore, what does not get produced? The product. Product is not produced. So we have effectively shut down that enzyme through competitive inhibition. Okay, let's look at non-competitive inhibition. How does that work? I'm going to draw another enzyme here. Okay. And this time, I'm going to have a chemical bond here to the allosteric site. I remember that pocket right there is called the allosteric site. I drew it in a previous slide. And when it binds there, here's what happens to the active site. The active site used to be open, but now it's going to collapse. All right. Once it's collapsed, there's no way the natural substrate can get in there. It's blocked. And because it's blocked, no product is produced. All right, so in both cases, you block the production of a product, right? You block that, you block that, but you did it through slightly different mechanisms. One through binding to the active site, the other one binding to an allosteric site. It also turns out that non-competitive inhibition, binding here, has a tendency to be difficult to remove. Like that guy here doesn't come off so easy. And for that reason, it tends to be a poison, something that permanently shuts down the enzyme. On the other hand, competitive inhibition over here, that's not necessarily permanent. This competitive inhibitor can fall out. And when it falls out, the active site is open again, substrate goes back to becoming product. So non-competitive inhibitors have a tendency to act like poisons, permanently shutting down enzymes, whereas competitive inhibition tends to be reversible, while this one is irreversible. All right. What about feedback inhibition? Okay, I've got a slide, a couple slides later that will make really good sense of that. So it'll look visually easier to explain. Okay, so in summary, here's the enzyme. Here's a really nice open active site. Substrate goes in, product will come out, right? That enzyme works normally. Or you might have a chemical that can fit inside of the active site, successfully blocking it so that this substrate can no longer get in. So this would be a good competitive inhibitor example. Or you can have a non-competitive inhibitor binding to a separate area on the enzyme, the allosteric site, which collapses the active site so that the substrate cannot get in there. Even though nothing's in there, the shape is different, substrate cannot go in there. In both cases, you prevent product from being made. All right, sometimes our cells want to control the enzyme. They don't want the enzyme producing all kinds of product all the time. So how do our cells regulate enzymes so that they're not overproducing product, which would be inefficient? Okay, let's look at a negative feedback loop, right? Negative feedback loop. All right. In this example, we're looking at a metabolic pathway. Got to work on that penmanship. I'll get better. All right. So the starting molecule is red box A. Enzyme 1 handles red box A. Notice that the active site, shaped, very boxy. That guy's going to fit in there nicely. And what is enzyme 1 going to do with red box A? It's going to turn it into a product. And the product is going to be blue triangle B right here. And then that blue triangle B is going to fit into that enzyme. And notice how the active site of enzyme number 2 has the shape of the triangle. This again showing the specificity of enzymes. Enzyme 2 works on B, not on A. Enzyme 1 works on A, not on B. What does enzyme 2 do to blue triangle B? It turns it into oval C. Enzyme 3 takes oval C and turns it into star D, green star D. All right. This is the final product of this metabolic pathway. The goal the entire time was to turn red box A into green star B. But you couldn't do so unless you went through a couple of intermediate steps, right? The intermediate steps represented by B and C. All right. But what if the cell finds that, wow, we've made a ton of this product? Well, that product can then come back around, attach itself to the original enzyme 1, and look at what it did to the active site. It shut it down. So that now a box A cannot fit in there. And if A cannot go into enzyme 1, then there's no B. If there's no B, there's no C. If there's no C, there's no D. And we have successfully regulated the production of that green star product. That's a negative feedback loop. And you should understand that that's mostly for efficiency's sake, right? And to regulate the production of certain molecules. All right, you should be able to explain to me the advantage of feedback inhibition at this point. Okay, we're not done. There's other ways to inhibit enzymes. You can inhibit enzymes through other mechanisms that we haven't described. And to understand these mechanisms, you would have to remember that enzymes are made out of proteins. And because they're made out of proteins, they can be denatured. Do you remember how we denatured proteins? We denatured them by use of acids, bases. We use salts, right? Even sugars could do it. Heavy metals. Heat, right? High heat will denature proteins. And things like radiation, right? Radiation will destroy a lot of things, including proteins. So we could also denature proteins and prevent the enzymes from creating a product. It turns out we can also enhance the activity of enzymes. So we can discuss enhancers, not just inhibitors. Easy enhancers to understand would be like minerals and some organic chemicals such as vitamins as examples of those organic molecules. So how is it that vitamins and minerals help your enzymes perform better? All right, so remember, enzymes have this allosteric site. Sometimes minerals bind here. Minerals such as copper, iron, zinc. There's bunches of enzymes that we rely on that need these minerals to be attached to enzymes. And in this way, the enzyme keeps a really nice big active site, and it makes it that much easier for the natural substrate to go in there. What do you think would happen if we took away the copper from an enzyme, or the iron from a different enzyme, or the zinc from a different enzyme? Well, then those enzymes would have their active sites collapse. And at that point, the substrate can no longer enter, and therefore what's not produced? Your product, right? So we need minerals. If you were wondering, why are vitamins and minerals so important to us? There you go. That's one really important reason. Enzymes. Okay. And just like copper, iron, and zinc can fit in there, so can vitamins. So if you're not getting enough vitamins, you'll shut down these enzymes, and you're not going to get product, and maybe you're not going to get the energy you need because you're not breaking bonds that release the energy that you need, right? Okay, so at this point, I expect you to be able to tell me how you can inhibit enzymes, right? Give me all the various ways in which you can inhibit enzymes or flat out stop them from working. And you can also tell me how you can make enzymes perform better. All right, here's an example of how you might have inhibited an enzyme for good reason. Things like pain and inflammation are produced by a group of chemicals called prostaglandins. Originally discovered in the prostate gland, so that's where the term comes from. Later we found out that they're everywhere else too. Prostaglandins are produced by a series of enzymes. So you'd have enzyme 1, enzyme 2, enzyme 3, enzyme 4, And eventually, all of those enzymes would contribute to the production of prostaglandins. And when you produce them, you get pain and you get inflammation. But what if you don't want pain or inflammation? Well, then go back and block one of those enzymes, any one of them. As long as you block one of them, right, you're not going to get prostaglandins. You can block the last one, or you can block the first one, or any one of these, right? And what's one way that we do it? Ibuprofen. Ibuprofen will block them. And that will get rid of the pain and the inflammation. Okay. But does taking ibuprofen one time permanently relieve you of pain and inflammation? Or does pain and inflammation come back so that you have to take ibuprofen at a later time? Yeah, pain and inflammation come back, and so you'd have to take ibuprofen once again. So what does that tell you about how ibuprofen likely works? Is it a reversible or irreversible drug? Is it a competitive inhibitor, or is it likely to be a non-competitive inhibitor, right? Yeah, you should have an answer. You should have some ideas. I'd like to hear from you. If you email me, that'd be great. All right. And it turns out that it's not just in the case of pharmaceuticals, pesticides, right? Pesticide manufacturers have also figured out ways to target vital enzymes on certain insects, enzymes that they have but that mammals don't have or that we don't have, so that it's safe to use these pesticides on these insects because they're not going to harm us as mammals. Unfortunately, in the insect world, there are also insects that we don't want to harm, like bees. And so using some pesticides to target one bad insect can sometimes have an adverse effect on the good insects, like bees, unfortunately. And still, others, of course, can do something a little more sinister, right? Okay, so do you remember what might determine if an enzyme is reversible or irreversible? You might want to go back and check the slide on competitive versus non-competitive inhibitors.